Okay, you can start.
Okay. Hello, everyone. It's a pleasure to Pharmaceutical Sciences School of the Ribeirão Preto to have here as participant of this very special lecture. Our speaker today is Dr. Ada Yonath. She dedicated her career to understand the complex architecture of ribosome. In 2009, Dr. Yonath received the Nobel Prize in Chemistry for studies of the structure and function of the ribosome. Driven by the desire to understand how the genetic code is translated into proteins, Dr. Yonath has developed a pioneer technology in crystallography. Her insights have also led a better understanding of the mechanism of drug resistance and paved the way for structure-based drug design. Dr. Yonath is currently working in the Martin Allen Kimmel Center for Biomolecular Structure and Assembly at the Weizmann Institute of Science in Israel. Today, she's going to tell us about the next generation of antibiotics. Dr. Ada Yonath, once again, thank you for accepting our invitation and gracing the occasion. Ladies and gentlemen, I'm giving Dr. Ada Yonath.
Out?
Yes, I can hear you now.
Okay. So I will share. Okay. I cannot share. It says that the host disabled me. Can you let me share?
Yeah, just a second, we'll figure out how to do this. Flavio, please.
Yeah, I activated, just activated.
You activated it?
Yeah.
It's almost there, I still can see.
Yeah, but I cannot start.
Flavio is our expert man. Flavio, what she should do?
Let me
Start again.
Yeah, maybe. Yeah, it started. It said start sharing.
It doesn't take the stop share.
Just a minute. It's not saying that it's possible to share what they are, because it's in my screen is saying that you are sharing, but it's not showing anything here.
No, it's all black.
Yeah, strange.
Yeah, sure, it's on black.
Maybe, let me see.
Do you hear me?
Yeah, I'm hearing you. Okay.
Yeah. I'm also hearing you, but I see nothing.
Okay, so please try again. I just give you I give you the possibility to share. So
Okay, I will try again.
Now it's
Perfect.
working.
Now I can hear.
Yeah, but I cannot get it into presentation mode. I have only the first page.
it's not going to the presentation mode, no?
It doesn't agree.
Yeah.
Well, maybe I can do something else.
What she should do, right?
I'm trying to check.
Okay, okay, I got it.
Perfect.
Hopefully it's perfect, but at least it's okay for the first slide. So I want to talk to you today about next generation environmental friendly antibiotics, but also about origin of life, which is a very new discovery that we made, and I want to connect between both. So origin of life is based on structural conservation. Everything to do with origin of life is detected by structural conservation. Next generation and environmental friendly antibiotics is based on conservation, but more on species-specific structural features. So let's talk about both. First of all, the central dogma, I think you all know it, DNA has the genes. They are being transcribed to messenger RNA and then translated to proteins by ribosomes. This is the way it happens in every living cell. How does the ribosome work? In a few words and an easy to draw story. Ribosome has two subunits, a small one and a large one. The small one hosts the messenger RNA, it means the code, and the large one hosts the peptide bond formation center. tRNA brings the amino acid. I think you all know what is tRNA. Each amino acid has its own tRNA, and it's determined by the sequence of the free nucleotides of the tRNA, which should be matching the free nucleotides where it's going to be connected. So there are 3 sites for connections. This will come here. When the blue will go to the orange. This is called the A site because amino acylated tRNA is here. This is the P site after peptide bond formation. From here, the protein goes out. And then after the bond is being made, the P moves to the E site and then exit the ribosome. So there are 2 subunits, small and large. The message is being read in the small and the bonds are made in the large. Ribosomes, there are many of them in every living cell. You can see here many ribosomes, one after the other. You can see here that they can even be arranged in a circle. And every one of them works according to the scheme I just showed. Highly active mammalian cells, for instance, in the liver, may contain up to 6 million ribosomes, even in small animals. Even bacterial cells contain between 50 to 80,000 ribosomes in each cell. So when we had got the first structures back in 1099, and when we got the first structures, immediately we understood, within a few months, we understood how ribosomes work. And we made a little movie, little clip. So you see here, messenger RNA reaches the small subunit. First, it binds to the small. And this binding is controlled by initiation factor, in particular initiation factor 3, this blue thing here. Now messenger is in its path in the small subunit, everything is fine. So the first tRNA can be brought in by another initiation factor, number 2. This is this blue thing. So the tRNA bound to subsite P, peptidyl transferase, is sitting here. You can see the amino acid. This is this little blue dot that you can get a knowledge of the size. And then the large subunit can reach, all the factors leave, and the large subunit makes its place in its position, accommodates the P-site tRNA, and immediately lots of tRNAs are being brought by initiation factors from the cell according to the sequence, to the sequence of the DNA. And they are get in and out, ribosome helps, in, out, in, out. And we took away the large subunit. We left only the pieces that have to do with making proteins. So here, the sequence is being read, and here, the protein is being made, and at the end, this process continues, and at its end, the protein will come out from here. Sometimes already gets its final structure alone, sometimes with chaperones. And when there is the last, somehow it stopped, but the last amino acid will be released by release factors. So what actually we saw is small subunit, large subunit, tRNA binds to both of them, here to decoding, and here to peptidyl transferase. It means peptide bond formation. tRNA is this yellow thing. Messenger will come out from here and go around and being read by the tRNA. Oops, sorry. Okay. So what we see, if we look at it from the other side, this is the small subunit, the large subunit, 3 positions of tRNA are reading the sequence from messenger here, and the PTC is where peptide bond is being made here. And I will describe it a bit more in a minute. So essentially, the ribosomes are made of many RNA chains. And it's the noise coming from me. In many proteins, each protein is shown in a different color, whereas the RNA in gray. And you see the small and the large subunit in bacteria. The decoding happens in the small one, and peptide bond formation is made in the large one. This is what we found already by just knowing the structure. Peptide bond formation is shown here. A-site tRNA, 2 P-site tRNA, they are making the bond here. I won't go into the chemistry more than here, but I'll come back to it. If we look at peptide bond formation positioning, in all ribosomes, it happens in the same way. Ribosome here is in gray, A-site tRNA in blue, and it will stay blue, and P-site tRNA in green. Peptide bond will be made here. And if you look at it, there is no catalytic intervention from the ribosome. It's just the two tRNAs with the amino acids or the protein bound to them in the correct stereochemistry to make a bond. And the ribosome here is in gray. And that's correct for all ribosomes. Doesn't matter if it's a small bacteria or a huge liver cell. And we can show it when we look at the way several tRNAs or several substrates are bound to the active site. And these are substrates that were obtained in our lab and at Yale. And you can see, maybe I will try to hold. I can. And you can see that both sit in the same way. It doesn't matter if they are very long, like this one, that has a big piece of tRNA, or this one that has already growing chain. They all sit in the way that allows peptide bond formation. Just to remind you that all tRNAs are made of a L-shaped molecule with anticodon loop at the other side of the part that binds to the amino acid of the peptide, which is called the 3' end, that is fully conserved, always CCA amino acid of protein, always. And this part binds to the active site of the ribosome. So here we put together active sites from 3 different ribosomes, from 3 different bacteria, obtained in 3 different laboratories. And I must say we didn't talk much to our competitors, and they didn't talk much to us or between them. Nevertheless, we got almost the same location of not only of the whole chain, but of each base here. And if we superpose the whole active site, you can see that it's beautifully overlaps one on top of the other, from the A region in blue, the P region in green. As I said, I will keep it. And that's correct everywhere, and it shows internal symmetry. The internal symmetry is really very important. I'll come back to it. But first, I want to show you that the tRNA from the A site goes into the P site within a frame made by the ribosome. You can see the frame here in gray. This is the way it goes. There is not any additional volume here. Everything is taken and very fine designed. So if we look at it from top, the region here is now shown from here to here, from A to P. This is the gray is the ribosome, and 2 nucleotides are shown here, 2585 and 2602. And I'll come back to them. And when they go from A to P, the bond is being made. Now we collaborated with Lumasa in New York, and they calculated when the bond can be made already, when there is the transition state position, and they found it here. And they also showed that all this way is important. So here is the transition state within this position. And I hope that I can show you what happens. So tRNA, the A site here is in the blue, and in the P site, it's in the green. And the way from blue to green is shown here. And the interactions with the ribosomes are shiny in yellow when the tRNA interacts with them. But there is not any reaction that the ribosome is involved in, only in positioning. Actually, a Japanese group of Suzuki wrote to me one day and said he has several nucleotides that he showed that are important, but they are not mentioned in any paper. Actually, people found biochemically that these nucleotides here and these nucleotides here are involved. Nobody looked at the nucleotides on the way. And Suzuki didn't know what we are doing, but we found that indeed the nucleotides that he found are important, are indeed important. They are sitting on the way. So what I want to show here not only is that Suzuki found it, but also that each atom here in the path from A to P contributes something is important. You remember there was a nucleotide earlier in the middle, 2602. This is this nucleotide, and you see here many of its structures. Each of them is dictated by another antibiotic that binds to the ribosome, and I'll come back to the antibiotics in a minute. But it has a lot of space for motion and a very intelligent motion, very intelligent, I mean, from the point of view of saving energy. So we also think that it may propel the whole rotatory motion. So if we look at the position in the two-dimensional make of the ribosome, if we look at the position of the P site and the A site of the what until now I showed you the pocket where proteins are being made, peptide bonds are being made. They are sitting in a place within the whole sequence. I'll show in a minute. They are about 90 and 90, between 70 to 90. Depends how you count it. Nucleotides on each side. And as I showed earlier, they have very high symmetrical positioning. So here it is the same thing, but with a little bit more of the two-dimensional representation of the ribosome. The sequence conservation of this whole nucleotide, this whole pocket is showing here. You can see that sequence conservation is very high. Most of it is over 50%. Very few are less than 50%. But what's interesting is that there are many that are 100% conserved between the two sides. There is no internal piece of ribosome that shares this high conservation. Okay, so in order to see what happens there, after we identified the pocket, we thought that we better make a pocket in the lab that imitate one of the pockets that we found in bacteria or in higher organisms, or several of them. And we wanted to see if we produce this type of pocket in the lab, we can also produce a peptide bond. And for this, we use to show it, we use the fragment reaction. I just want to tell you that what I'm talking about now, the experiments to show that we can produce this piece of the ribosome and make a bond took us over 10 years. So first we produce the pocket, just the pocket, and then we try to see whether it can make the reaction. Just to remind you, the reaction itself is shown here from the A site to the B site. So, as I said earlier, the ribosome is only orienting the substrate that makes the peptide bond formation. It obeys two-fold symmetry, but there is no sequence symmetry. The sequence is different in both sides, and we think that this shows that one side is entering the tRNA and the other is expelling out. So if we look at it, we think that this is the missing connection between the frame components in these positions. It means the blue and the green are shown here. And these positions are not bound chemically, but still the structure is very stable. Oops. So if we look at all what I told you until now, we call this internal piece, the blue and the green, the proto-ribosome, because we think it was before ribosome. It's made of two parts, blue and green. Peptide bond will be made in between. It is the same in many, actually in everything we checked. Here it's a superposition of many structures. And from the side, it looks like this. So this essentially, you can see here which sources we used in order to create this pocket. It means we show it the universality of the pocket. And as I mentioned earlier, we tried which constructs make proteins, the capabilities of the constructs. And you can see that not all of them can make peptide bonds or proteins. Only part of them, and I think, to remind you, this is the reaction that we had. And as I showed earlier, together with the reaction, only 5 out of 13 what we call proto-ribosomes. Proto-ribosomes are the blue and green that I showed earlier. That's essentially a name we gave. All peptide bond formation is made only by 5 of them, although we got 13 as pockets. So just to show you, hopefully it will show how this proto-ribosome looks when it rotates. Together with the substrates. Oops. I try, wait. Okay. I just want to say that in other labs, they also talk about the ancient ribosome. And for instance, in Atlanta, peeling the onion, they didn't go on, but they agree with our suggestion. So this is a piece that is fully conserved in every and all ribosomes. So let's go now to the next part, which is next generation and environmental friendly antibiotics. And I will combine the two of them based on the structure. So the the protoribosome is the same in all ribosomes. The positions of antibiotics, some are and some not, and I'll talk about it. So right when we got the structures, we determine the structures of the ribosomes with, wow, yeah, okay, with antibiotics. So we can see here, again, this yellow thing, very clever, sitting at the path of the messenger RNA and become antibiotic just by not letting the messenger bind. It's not allowed to use again because it is not very specific, but tetracycline that you see here is also very small. It sits in the position of the A site, and it's extremely specific and very useful, tetracycline. The third one that we show is erythromycin, the first one of ribosomes that was discovered right after penicillin, actually. And it sits at the beginning of the tunnel through which protein will come out, just at the end of where proteins are being made. So all antibiotics that we know paralyze the ribosomes by binding to their functional sites. And I can show here several of them. In the movie you saw one that sits in the active, in the path of the messenger, one in the active site, and so on. So the functions that are disturbed by them are decoding, hinging, it means the motion of the small subunit, tunneling, and bonding. So, as you could see, most ribosomal antibiotics bind to ribosomal functional sites, but they are highly conserved in all ribosomes and perform the translation in the same way. Nevertheless, mandatory for clinical use is the distinction between the patient and the pathogenic bacteria. So if they bind to conserved places, they good for nothing because they will also disturb the patient. Well, they enjoy very few differences. So you can see here environmental at the entrance to the tunnel is the large subunit, and you look at it from top. And if you look at it from the side, here is where it sits at the entrance to the tunnel. tRNA, P site tRNA sits here, the PTC is here, and it sits here at the entrance. What happens? From erythromycin, several sons and daughters were made, all bind to the tunnel at the same position. They are called macrolides. They are pieces of, they are compounds that are coming out, or derivatives of erythromycin. And they all have something to do with this nucleotide that I showed you earlier, that we think has to do with transfer A to P site tRNA, 2602 and 2658. And 2058, sorry. So if we look at it, this is the way 2602 moves with different antibiotics. And this is the position when the antibiotics bind. I think I showed it to you. And the differentiation between pathogens and patients is based on subtle differences, very small differences. You can see here, this is erythromycin, and A in position 2058, everything is fine in pound. In eukaryotes, there is G instead of A. That's the only difference. And they don't bind, it's too close back. So this is the discrimination between small, not only small, between bacteria and patients. But about how antibiotics work, there are many papers, and not all of them are talking about the biggest problem of the 20th century, resistance to antibiotics. This is one of the most severe problems in modern medicine. And study on the health of people who had never been exposed to Western medicine or diets and live actually in South America, well, I think, I'm not sure it's Brazil, but it's north of Brazil. It showed that even people that never saw antibiotics or medicine had already antibiotic resistance. They have the genes for antibiotic resistance, which shows that the resistance is made naturally, doesn't need the antibiotics to invoke it. So this shows that resistance to antibiotic is a basic process for the survival of many microorganisms, regardless of their exposure. I just tell you all the problems of resistance. And also it suggests that microbes have long evolved the capacity to fight toxins. For them, antibiotics are also toxins and prevent drug resistance. That we are interested maybe harder than what we think. And indeed, it's hard. And it becomes a colossal health threat in the 20th century because there is no resistance to any antibiotic, antibiotics. And it seems that we will soon revert back to the pre-antibiotics era, during which diseases caused by parasites or by simple or severe infections were almost untreatable, as it was before antibiotics. We are going back there because of resistance. So we are essentially reaching post-antibiotic era, as it was called by the World Health Organization. And consequently, the World Bank estimated that up to almost 4% of the global economy will be lost by, now they have all types of years that they give to it, but relatively soon because of resistance to antibiotics. But very few new antibiotics are under development. Most large drug companies have stopped attempts for creating new antibiotics. And this is owing to the huge mismatch between the social value of new antibiotics and the high expense to produce them. So they are making more antibiotics, but copying the old ones or really changing them, but not new. So, as I said, most pharma companies stop searching for new antibiotics, owing to the expectation for resistance that's also a reason. So you can see here the increase of drug resistance on the left between 1990 and 2002, 12 years. There are here three different bacteria that develop resistance. And here you can see the number of new antibiotics from 1984 to 2014, 30 years. And this is all what the companies made. So there is still a decrease of this. What they are doing usually is taking macrolides like erythromycin and changing them, making them more suitable, no question about it, but just using natural wisdom for making new antibiotics. And the question is, do we think that we can combat resistance to antibiotics? So we think that it's unlikely because bacteria, we call it want to live. They will find ways to resist. And they are essentially in this respect cleverer than us. Nevertheless, we try to contribute toward controlling or combating antibiotic resistance. And we had some modest contributions which I want to describe now. So until recently, almost all available crystal structures were of pathogen models. These were found suitable for illuminating common modes of antibiotics action, but do not explain specificity of pathogens. Our concept for fighting or controlling the bacteria's wisdom was identifying pathogen-specific structural motifs and using the ribosome because it is such a useful target for antibiotics. So first, we compared, we determined the structure of several pathogenic ribosomes of pathogens and compare them. And we have many lessons, and I will describe only very few. So the general, all bacterial ribosomes are similar, but I didn't say that they are identical. They are just similar. Lesson 1 is that it's possible to increase antibiotic activity and make more potent antibiotics specific to some of the pathogens, and I will not show it now, but it's possible. Nobody said it. Lesson number 2 and 3 I will skip. Number 4, this is beyond my expectation. It actually talks about novel antibiotic binding sites, those that were not seen before. And I want to describe this. So what you see here is the non-pathogenic ribosome, large subunit, in gray, and pieces of the pathogen that are more than the non-pathogenic bacteria. Additions, very few additions, but they are there. And they have structures. So we focus only on those that are on the surface. Let's look, for instance, on this one down here. You see that both bacteria have same features. Oops, sorry. I can do my mouse. Here, they have the same features here and here and here, but this part is only of the pathogen, and it has a structure. It is on the surface is exposed. So we think that it can be a potential binding site for antibiotics. So if we look at the whole structure, the whole large subunit, there are several potential binding sites in addition to the first one. So essentially, so far we identified 25 new potential sites for the design of advanced antibiotics. And we designed antibiotic or binding compound to each of all 25 potential sites. And 16 of them were found to inhibit protein or ribosome function. So this means that so far bacteria did not assign vital roles for those. So for now, until bacteria becomes cleverer, they can be exploited for the design of advanced degradable antibiotics. Degradable. Why is it important? From the point of view of environmental problems. If they are degradable, they can make them degradable because most of the natural antibiotics are not. So we can control also the environment. So then we looked for positions, and actually we don't need structures, or we need structures only later. We have here so many so many bacteria. Most of them are involved somehow in diseases. And we put one on top of the other, and we found positions like this that are specific to a specific bacteria. And then we made to it a compound that binds to it. And I want to show you what I mean. This particular position here is shown here. So everything in the neighborhood is the same between normal and pathogenic bacteria, but this is specific, so we can use it as a potential new species-specific target. We have several like them. You can see one that I highlighted in green, but all those that have colors have no contact. It means they are exposed, and we can even show it nicer, and we can make antisense to them. That's all shown in Staphylococcus aureus. So you can see here, and here, and here, and here, and even here, which is much nicer, this blue line is going between unmatched pieces. It means available pieces. So we made the species-specific oligonucleotides, antisense oligonucleotides, and you can see how they reduced the IC50. It means the amount needs to stop the function of the pathogen. You can see even more here. And those antisense we can design the way we think is correct, the way the degradable, not disturbing others, and so on and so forth. Specific, most important, specific and degradable. So we can use normal antisense, but also double antisense, like here, which is less strong, but nevertheless still works. And we can do it also on really specific positions, like in tuberculosis. You see all this part is unique. So we could hit it here, here, and here in these positions. So maybe we will have soon something against tuberculosis. essentially addition to the previous slide is shown here, showing more specifically where we can bind here and there. So this was to look for specific pieces, but we can also hamper functional motions. Like we showed that helix H68, it doesn't matter the name of it, but this is this helix, has two positions. When the E site tRNA exit, it opens up to allow the exiting, so we could block it here. And this will hamper functional motions. And we work on this one now. And this is too complicated, let's go on. But we can also predicting target site in different bacteria. And we are doing this too. Sorry. And we can even look at multi-resistance pathogens. Recently, we worked only on structures of parasites, ribosomes from parasites like Leishmania. And we found there also a few positions that are suitable to become target site, but we are not going to explain it now. I just want to show you how good the structure is, even a piece of it. So before I finish, this is my group. Several of the people here have already left, but I want to highlight Yonath. Yonath, she is really the head of this group. I am there just for the picture. The protoribosome and its activity in making peptide bonds was done by Tanaya, but it was identified by Ilana and by Chen. Giuseppe found the motion. Judah found the faraway positions, and Gil is designing the antisense, the probably good future antibiotics. Tamar had a birthday. Tamar came to us 18 years ago for 10 weeks, and she's still around. She had a birthday when I took the picture, and she made a cake. You see the cake? This is the ribosome. And it says that in our group, ribosomes are considered sweet. I also want to thank the Weizmann Institute that let me work, and the Max Planck that for 16 years set the lab for me, and to you for listening. Thank you.
Thank you very much, Professor Anna. It's a really wonderful lecture. It's really a lot of research area to be developed. So it's just perfect, your lecture, and I'm sure that it will be an inspiration for young scientists to continue to work in this area and maybe discover new antibiotics using this technique. Well, I know that probably have a lot of questions, but maybe one or two if the audience is interested to ask something.
Roberto, can I ask a question then?
Yes, please.
Okay, Professor Ada, thank you for the fantastic talk. Always a great inspiration for us. I actually have a lot of questions, but one specific question is regarding the identification of the binding sites, comparing the pathogenic and non-pathogenic bacteria. And you mentioned that out of the 25 potential binding sites, 16, I guess, were validated. So, and there were like, I don't know if I can say or use this word, but could be allosteric sites, or I don't. So my question is actually that, I mean, they were validated as important for modulating the activity. So how? I mean, is in dynamics of the structure, how they are, cross-talking with the binding sites, the well-known binding sites from ribosome. Thanks.
I think we do according to antisense, just chemistry. Why are they important? They are not sitting in the ribosome. They are on the surface of the ribosome. And we think we are now going one by one to show which contacts it makes with the cell, with proteins or components of the cell that are important for cellular growth. So it doesn't have to be only the ribosome, it's also the ribosome and within the cell.
Thank you.
Well, thank you very much, Chris, for your question. If anyone wants to ask a question or maybe send an email with that question? All right. So I will ask Flavio, please, could you just say some words and finish the lecture? Thank you very much, Dr. Ada, again, to attend my invitation, and I hope to see you in the future, maybe in Brazil.
Thank you. And if any of you people want to come and work with us, if they are good, we will take them.
It will be very good, of course. And I will take this opportunity to thank you again, Professor Ada, and thanking Roberto too for making this lecture possible. I'm sure all the students here, and we are seeing lots of messages in the chat, congratulating the lecturer, of course, and the awesome work and awesome presentation. We have lots of people watching also in the streaming, so it took a lot of attention from the community, and I'm sure it will give us a lot of results from this first interaction. And I have to say that was a really honor again to receive you, and it's very important for us, for the students that are future researchers, that having this inspiration coming from you, for sure will motivate them to make their best, and to make them to be the best they can. So that's what we can, that's the thing we can do. So thank you for the audience. So I invite you all to go to the graduate program in pharmaceutical sciences webpage and in social media, and share the message of this talk and all the initiatives that we have been doing for developing the post-graduation in Brazil. Okay, and this is one of the efforts that we are doing. Thank you again. Hope we can see you soon in the future, and be well. And
thank you.
Flavio,
thank you for the invitation.
Juliano?
Flavio, may I just, hi, my name is Juliano Kozowski. I'm a colleague of Flavio in our faculty, coordinating this postgraduate program. Yeah. And I just would like to thank you for our time, for excellent lecture. It was very inspiring to me, and I'm sure for all the people that attended. Yeah, and really a great pleasure. And I just was thinking, and then maybe it was just finishing the activity, but I was thinking about, you mentioned this small amount of investment of industry, pharma industry in new compounds and new antibiotics. And how would we solve this problem? Because apparently, as you mentioned, bacteria are being cleverer than us in this moment. How to solve this problem?
I cannot solve all the problem, but I can contribute what we are doing.
Yeah, but in the pharma industry, people like new compounds or new modes of actions or things like this could be a way to launch new compounds, and in some way to fight against this problem, because we know that antibiotics are quite cheap, yeah, when compared to other drugs. Yeah, maybe this would be a problem.
It's a problem for us. But I guess that if the problem continues, if the resistance problem continue, the companies will be able to charge more for antibiotics, make the price higher.
Yeah, that could be a way. Yeah,
maybe
because
tell them.
Yeah. The way they work. Yeah. Oh, well, thank you very much, Professor. Really good pleasure to have you.
Thank you for inviting me.
Flavio.
Okay, thank you, Juliana. And
Okay.
now it's time to say goodbye. Bye-bye.
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 Bye. Have a nice day.